Okay, so today I'm gonna do a PCR in my lab for my project. To be brief and to oversimplify things, I'm trying to build a mammalian expression vector, which is a plasmid, basically a circular piece of DNA that I'm trying to insert into a mammalian cell, first into the cytoplasm, then into the nucleus, of course, to make the mammalian cells do express a gene for me, which of course is inserted into this plasmid. So this would be my gene of interest, and I'm going to try to make the cells express this gene. Of course, there's a lot of hard work going into this, and for now, I just need to do a PCR. What I'm trying to do actually is just amplify a bit of DNA from another plasmid, which is called pcDNA 3.1, which is a plasmid that I previously modified. So I'm just trying to amplify a piece from here to here. So this fragment will act as a brick or as a fragment that will go into building my mammalian expression plasmid, right? Uh, so regarding the PCR, I'm not gonna talk about designing the primers. I'm not gonna talk about ordering primers. I'm not gonna talk about hydrating them because once you get the primers, you're not gonna get them in liquid form. Usually, you're gonna get them dry, and you're gonna have to add PCR water to them, and you're gonna have to make a stock concentration, and from this you're gonna have to dilute and make a working concentration for both forward and the reverse primers. I'm not gonna talk about picking your template DNA because, as I told you, this would be my template DNA. But first of all, before doing a PCR, this might come in useful for you guys just getting into, you know, practical experimental biology. You need to know two things. Basically, you need to know the reaction, the PCR reaction, what exactly is going into the PCR mix, and of course, in um, how much quantity, so what exactly are the volumes, and number two, you're going to need to know the thermocycling conditions. Basically, for this two, you have the protocol. So with each enzyme that you order for each polymerase, the supplier will provide you with a protocol which acts as a guideline for doing your PCR. For example, I'm using Q5 master mix 2x concentrate from New England Biolabs. So I know from their protocols that for a total PCR reaction volume of 25 microliters, I'm going to need to pipette the following. So I'm going to need to pipette 12.5 microliters of 2x neb master mix. Why 12.5? Well, because it's 2x concentrated. So for a total reaction volume of 25 microliters, I'm going to have to put in half of that. So 12.5. But first, what exactly is a master mix? Well, a master mix is basically a polymerase that contains all of the buffer and necessary reagents to do a PCR. So it also contains, the master mix also contains, besides the polymerase, it also contains magnesium, a buffer, and of course the NTPs. Besides the master mix, I'm going to need to pipette in my reaction 125 microliters of forward primer. I'm talking about the working concentration here. I'm going to have to pipette 125 of reverse primer, again working concentration. I'm going to have to pipette my template DNA. I'll pipette 1 microliters of pcDNA 3.1 minus BSE1. So my template here. And to get to a total reaction volume of 25, I'm going to need to add PCR grade water. So according to my calculations, I'm going to need to add 9 microliters. Okay, so this in total will equal 25 microliters in a PCR tube, of course. Then I'm going to have to put the PCR tube into a PCR machine. Right now I'm going to use a MINI PCR machine. I'm going to show you exactly how it works and how you need to program it. So I'm going to need to program it with the exact parameters that are recommended in the enzyme protocol. So I'm going to need to program it with the exact thermocycling conditions for my particular enzyme and my particular DNA. For right now, let's go into pipetting. Okay, now let's get pipetting. First, I'm going to need a tube. Just label it P from promoter. Okay. So first I'm gonna add the water. This is 9 microliters. This is my PCR water right here. So 9 microliters of water. And then I'm gonna add the master mix. This is 12.5. First of all, I'm gonna add 6.5, then add another 6. Alright, so here's the master mix. I don't know if you can see it. It already has polymerase and buffers and DNTPs in it. Okay, so let's add 6.5. Then another 6. You should keep, ideally you should make the reaction on ice probably, but yeah. Okay. Next I'll add the primers, because I don't have 125 microliters on my pipette. I'll just add 1.3. Just let me give these little guys a little spin. Okay, so we'll have the reverse primer. And then the forward one. You can see this is just a tiny amount of liquid. And 1 microliter of DNA. This is my plasmid right here. tiny, tiny amount. So I'm gonna give it a little vortex and a little spin. So here this will go into the PCR machine. All right. Now let me show you the mini PCR. So basically, this is the mini PCR, right? It comes in a box with some power cables. Something like this. The box is nothing to look at, so I'm not gonna show you that. So basically, you just plug this right in. This is the cool stuff. This is the cycler. It's pretty tiny. I really like this because it's so easy to program and well, yeah, it's kind of cute. To open it, basically you need to press these two plastic thingies. Just open it, right? And this is how the inside looks like. So this is the heated lid. You don't have to add any mineral oil or something like that. It has this is the MINI PCR 16, right? So it has 16 positions. Okay. So this is the tube. I just pipetted it. I'm just gonna put the tube in right here in a position. Close it till it clicks. Then just tighten it a bit. Not too much. Just gonna plug it in. Right? And as you can see, it's searching for Bluetooth right now. Okay, so let's get to desktop and program the thing. So before starting the protocol, we need to look up the cycling conditions for the Q5 high fidelity master mix that I'm using. So this is the protocol right here. You can see the reaction setup and the cycling conditions. Right, so for the cycling conditions, let's open up the MINI PCR app. Here it is. Let's connect the device. Let's check out the library. Of course, I already have the programs introduced into the desktop app. There is also a mobile app for convenience. Okay, so let's edit the protocol. According to the recommendations, the initial denaturation should be 30 seconds at 98. Then we'll have a denaturation step at 98 degrees Celsius for 5 to 10 seconds. I chose 7 seconds. Then we have an annealing temperature, anywhere from 50 to 72 degrees Celsius. For this particular polymerase I'm using, the annealing temperature should be 3 degrees Celsius above the TM of the lower TM primer, right? I'm gonna use 64 degrees Celsius, right? Because I know the TMs of my primer, and one of them is basically 60 or 61 degrees Celsius, right? They recommend an annealing time of 10 to 30 seconds. Oh, sorry. Right, I chose 20 seconds and an extension. Again, this is polymerase dependent, the extension time. For Q5 is 20 to 30 seconds per kilobase for genomic samples, but can be reduced to 10 seconds per kilobase for simple templates. I have a plasmid and I know that my amplicon will be somewhere on between 700 to 800 base range. So I'm gonna do the extension for about 10 seconds. Multiply this by 30 cycles, and then you have a final extension at 72 degrees Celsius for 120 seconds, like in the recommendations here. Alright, so let's just click save and run. As you can see here, the lid temperature and the sample temperature, 51 minutes until it's finished, and the temperature will start growing and growing, and then the cycling will begin.